Okay.
Okay, so let's have a class and let's finish talk about the microscope. Microscope, then we can move on to talk about the staining technology. So we briefly gonna review. We talk about the bright field microscope. Microscopy. And we mentioned about several things like more important resolution power equals lambda divided by 2NA. And this, we wanted the lambda to be the smaller the better, because the smaller number will let the resolution power will be decreased. The value is decreased, the power capability is increased. And the second one is we talk about the magnification. We talk about the working distance. We talk about the oil immersion. And we also talk about the parfocal. So these are the things we finished to talk about bright field microscope. Then we move to talk about the dark field microscope. And the dark field microscope, which means the background is dark and you specimen, you show that. The reason is we mentioned about this very nice picture. There is a block here, so the light going through is perpendicular. It's 90-degree angle. And this is used to see the very special bacteria, which is spiral sheet. And the typical example is Treponema pallidum, and this one will cause syphilis. So this is what we talk about there. Of course, there's others, like Borrelia burgdorferi for Lyme disease, and we will mention later on. So this is something we talk about already. And today we're going to continue to talk about the next one. Okay, phase contrast microscope. This is something little bit talk about is physics. So phase contrast microscope. You need to know a basic physics behind this. It's kind of interesting. You should know about that. If I have a wavelength of a light, let's say this is a light wavelength. And I have another light wavelength coming. If it is a peak versus peak, what happened? It is dark. Okay, if I have a wavelength like this, and I have a two peak which is double layered, this will become brighter. Now, this is what our normal light going through the microscope looks like that. So, let's give an example. And we had a picture which is right here. Could be showing you something. So, let's say do it. I have this guy right here. Okay. Then I have this guy. Let's do it differently. Just to make sure you understand. Okay, this is a screen, let's say. So, if the wavelength going through condenser, it looks like this. Based on the physics, it's going to be dark. You're not going to see anything. Because the top overlapped. So what they do? These guys are very smart. It's a physics person. They put a ring here. This is called a phase ring. What's the function of the phase ring? They will let the light wave approximately 1/4 wavelength ahead of time. Sometimes a phase contrast microscope will be back 1/4 wavelength. Most of the time is 1/4 wavelength ahead of time. Therefore, instead of you have this, what it's going to be? It's kind of like that. See, that's going to show that. So, what you see? That means you're going to see some of the brighter area. That's what's a simple physics behind it. So originally, if I didn't put a phase ring, it's going to be dark. So I put a phase ring, let the wavelengths 1/4 wavelengths ahead or back, the light show that. And if you think about the screen, we move, it's going to be finally go here. So this will be more focused and you're going to see the image. That's what's a function of the phase contrast microscope. But why we want to do this? This is used to do lots of the eukaryotes. For bacteria, for microorganisms, if it is eukaryotes, sometimes it's difficult to do the staining. We will talk very quick. The bacteria, we could do simple stain, gram stain, endospore stain to find the structure. But it is difficult to do sometimes for eukaryotes. So we try to skip that stage. But we also want to see the internal structure, because we want to see in internal what it looks like. Therefore, we comes out with this very magic phase ring. Change the light wave. And this is what it looks like. So you could see this is amoeba. And this is perimycin. You could see briefly the internal structure, what it looks like, without even doing staining. So that's a very magic tool about it. And here, you could see the comparison between bright field microscope, dark field microscope, and the phase contrast microscope. This is the yeast, Saccharomyces cerevisiae. This is the yeast using for beer brewing. So when we manufacture beer, you need to add a yeast which is Saccharomyces cerevisiae. And it's very interesting. If you underneath those different microscopy we just mentioned, which one you would be like? Obviously, you like the middle one, because it's shining, it's bright, and you can see the internal structure there. And this is the one you just have an art picture, and this is the one too shiny. You see the inside is like white, but you can see the internal structure more clear using phase contrast microscope. So that's what is the function of the phase contrast microscope. Okay, next one is fluorescence microscope and the laser confocal microscope. Those two, basically, we can talk about together. There is something which is similar. The principle a little bit similar, so we want to talk about that. The detail, what are they? So fluorescence microscope. Fluorescence microscope. First of all, you should know what is fluorescence. This is light shining yellow color. Now, I don't know about how much you know about the diamonds. The diamonds, you should not have too much fluorescence. If you have too much fluorescence, which means the purity is lower. But microbiology, microscopy, we like the fluorescence. Some of the bacteria have a natural fluorescence color. Let's say a typical one, Pseudomonas fluorescens. They have it. And Pseudomonas is a sporage bacteria. Sometimes we can evaluate the sanitation impacts. So, you're using some of the tool, you do a scanning, you see the fluorescence color, that's not good. That means Pseudomonas is there. So fluorescence is very useful. It's a light shining yellow color. Fluorescence microscopy, there is a little bit of difference from light microscope. The energy resource is different. They use UV light or blue light as energy resource. Different. Now, you know that the wavelengths of the UV light and the blue light, they are shorter than just the light. Is that right? Sunlight, let's say. They're shorter than that. So, this is going to be more focused because resolution power value will be decreased. It will be more focused. The resolution power will be increased. So this is the second thing I want to mention. Third thing I want to mention is fluorescence microscope, you needed to do staining. But this staining is very special stain. We're using the chemical called fluorochrome. And the very typical commercial one we're using is called Debbie. Let me make sure it's spelled right. Yeah, DAB. It's a Debbie stain. What's the function it is? It can stain DNA of microorganisms. Now, why we wanted to do that? Because we could differentiate dead and live cell. Dead cell, most of the time, is red color. I try to use a red pen to do it. Yeah, the dead cell is red color. And the live cell, most of the time, at least the one that I did in my research, which is green color. Now, obviously, another thing we need to do, because it's going to be showing the color, so you have to set up wavelengths. Okay, so that's something different. Need to set up wavelengths ahead of time. Red approximately 70-10 nanometer, green approximately 550 nanometer. You have to set up it. Then you could do the observation. Okay. Now, obviously, there is a mirror there, which is dichromatic mirror, because they reflect short wavelengths, transmit longer wavelengths. Longer wavelengths, we don't like it. We let it go. And the more focused here, the short wavelengths go there, is at the bottom. And what it looks like. This is a picture of what it looks like. Look at there. Lots of these are red color, dead cell. You see those green color is live cell. This is a very typical, looks like it's mixed with microbacterialides. You see those cork cell there. And you also see those large-shaped bacteria, very long chains. So that's a streptococcus there. This is something very interesting, looks like here. It's like a streptococcus, it's a chain. These kind of color, it's a typical fluorescence color, light yellow color. If we don't do any of this thing, so we don't know the dead or alive, but it's beautiful. And this is means some of the green color, which is stained, it's a live cell for some of the UK use. So that's what's the example. Okay, so relatively brief. Confocal microscope. This is the one we call it laser confocal microscope. Okay, laser confocal microscope is a very expensive tool. How expensive it is, somehow 100K. Yeah, 100K, something like that. Somewhere 100 to 200K dollar. It's expensive. Why it is expensive? The laser. They need using an argon laser. Coming from a very important, a famous company is this. So Z710, the ID number for the argon laser. The laser will generate a more shorter wavelength. That's why it is more focused. Okay, this is number one, more focus. Second thing, laser confocal microscope is connected to the computer. They have that detector which is back and forth on the surface. So, this could generate 3D images. The 3D images is very important. X, Y, and Z dimension that could be used to detection one group of the bacteria. That is called biofilm. Biofilms is a very special group of the bacteria. We know there's a bacteria growing in the median on the food product surface. They also could be bacteria grow on those bench surfaces. If the bacteria grow on the bench surfaces, they got to absorb the liquid there. Based on the van der Waals force, they start to connect together. Going through chlorine sensing or chlorine sensing process, which means cell-to-cell communication, transfer some of the chemicals, rely on possible gram-negative, it's going to be different. Then some bacterial cell will be repelled, some will be condensation, and later on what's going to happen? They will generate a very heavy 3D matrix. This generates a big problem for sanitizing and cleaning. If a normal bacterial cell, we only need, we call it a vegetative cell. We only need, let's say, 200 parts per million chlorine. If it's biofilm, if it's biofilm, you need to use maybe 2,000 ppm chlorine. You're not even going to kill it. It really depends on. And this is a big problem for the food industry. Because you go to meat plants, on the processing surface, more or less you have some meat residues in. And those residues is a good energy and nutrition resource for bacteria to grow. So if the person did not clean it very good every day, what happened? If they fail to do sanitation standard operation procedure, the matrix will become heavy and heavy and heavy. So we need to see the dimension. We need to figure out how to do it. Therefore, we need a laser confocal microscope. Okay? This is something what I did in my research when I was working 12 years ago at USDA ARS, which is Agricultural Research Service in Beltsville, Maryland, not very far from here. The thing is very interesting. I didn't do 3D images, but I used a color spectrometer. This first picture is a biofilm on stainless steel coupon. You see lots of them is green color. The color spectrum on the x-axis, which means it's green, it's live. The second slide, what I did is that I'm using distilled water to wash it. I don't do any sanitizing and cleaning. You see, some bacterial cells start to die. The green is live cell. The red is a dead cell because I use the cytosine 9, which is very similar to Debbie stain. What you see here, the color spectrometer started to expand it, and will show you the green but lots of red color. Go here, which is I'm using chlorine. I remember using like 200 parts per million chlorine. You see lots of them are red. The bacteria died. So you see here the peak value of that red is very high. And they have a little bit survived the cells there. The green is still survived the cell. So the last one, I'm thinking about chlorine might be not enough. So I need to add some of the surfactants. Because the surfactants could let the chlorine working really well. They have a large exposed surface for bacteria to be killed. Then what it looks like? The brownish color. Well, 2 cells still survive. We just don't want it to be, make sure it's 100% killed. Is that too good? So the color spectrometer is right in the Z line, which means it's brownish color, which means all those cells are most likely it is injured. So this is an example, which is a color spectrometer used in the confocal scanning microscope. But I didn't do the 3D images because it's not necessary. It looks like similar when we did the comparison. Okay, so that is a laser confocal microscope. Okay, next one, we want to move to electron microscope. Just be relatively brief, not going to be too complicated. Electron microscope have 2 basic different ones, which is scanning electron microscope and transmission electron microscope. Scanning electron microscope, we just acronym SEM. Transmission electron microscope, acronym PEM. So, first of all, energy resource. Energy resource of an electron microscope, it's electron beam. The wavelength is much shorter. Second, magnification. Scanning electron microscope, about 7 to 12,000. Transmission electron microscope, more than 20 to 30,000. Could up above to 100,000. It depends. Now, what they are looking for? Scanning electron microscope. This is scope, okay? 3D image surface. Because the lens is on the surface, is back and forth. So we see 3D images. Transmissions, electron microscope, internal structure. This is a major difference. So we gave you some of the example. Transmission electron microscope and scanning. You see the basic structure there. You see the internal structure is more obviously for transmission electron microscope. And the scanning electron microscope, you can see it's most likely is a 3D images is on the surface. Now, why are we using transmission electron microscope? Because we need to know the internal structure. We need to know the cell wall, composed by peptide or glycan, the modified sugar and amino acids. We need to know the ribosome. Remember what's a ribosome for? It's for the RNA transmission. And the bacterial cell has a ribosome. Eastern modes also has it. We need to see the location of the nucleus because the DNA and RNA is the most important genetic information there. So that's why the transmission electron microscope is important. But for some of the eukaryotes, I don't want to see so detailed inside. I want to see the whole morphology, what it looks like. That is what scanning electron microscope can be enlarged to see it. And this is a very interesting slide. Tells you the range. So, scanning tunnel microscope. The resolution power, you can see the wavelengths, 0, 1 to 10 nanometer. How about our light microscope? Above 100 nanometer. So they're almost 10 times or 20 times more focused. And this is, you can see a different, the microorganisms and some of the chemicals stuff there. You see all these amino acids, hydrogen atom, protein is less than 10 nanometers. Poliovirus, HIV virus, somehow 10 nanometer. But when you go up, these are bacteria, red blood cell, amoeba. You can use these. You could use electron microscope to do the observation to see more clear structure, but light field microscope is okay because the light wavelength is small enough to make sure the resolution power is good to do the observation. So that's a slide we're showing that. This slide, in my other class, I draw separately on the blackboard, but here we just briefly going to mention. This is clearly a difference between light microscope and the transmission electron microscope. You can see what's the difference. So clear. Light field microscope, using light as an energy resource. Electron microscope, electron gun. This is the #1 difference. Light microscope, you have those condenser, ocular lens, objective lens. This is all made by glass. That's why we have a refraction, because air and glass, they are different medium for light to pass through, so it will cause refraction. How about the electron microscope? They have an electromagnetic. And more important, light field microscope is going through air, the light waves. This is a tunnel, which is electron beams. So you can see the whole system. You don't have any reflection there. And the last, light microscope, we're using eyes to do the observation. Electron microscope, you view on the screen. Okay? So that's a very nice slide to show you the big difference between light field microscope and electron microscope. And typically this user example is transmission electron microscope. And that have to be done in a tunnel. Okay, so we say it's a tunnel to generate a vacuum. Then why we have to need a vacuum? Because we don't want other medium to go there to bother in the light refraction. It's more focused. Specimen preparation, there are two things I want to mention. The electron microscope is a sample. Let's say this is a sample. You need, first of all, is dry freezing. You need to very fast to dry freeze it. We could talk about this a little bit. Dry freezing is important. What happened? Not sure you understand about the chemistry behind it. If we want to draw it, a curve looks like there is, if this is a Pascal, okay, so this is this is a 3-layer, it looks like that. For liquid, this is we call it a sub-limitation line, because this is a vapor. This is solid, like ice, and this is liquid, like water. If you have a solid products at a certain temperature, this is actually T, temperature. The Pascal or using Miller mercury lens to measure it. This is a 5. Very interesting. 0 degrees Celsius temperature. If the temperature is low enough, as long as I make sure there's equipment, the pressure is less than 4.8 millimeter mercury column, the solids will directly become a vapor. That's called sublimitation. And that is the principles of dry freezing. So, basically the specimen, you have to do a dry freezing. Make it being very frozen, immediate. Because we have to slice it. Then we slice it. And then you're going to see some structure like this, some structure like that, some like this. What we do? We have to coat it with heavy metal. We don't say stain. Coat it with heavy metal. Then it will generate a large area, we call it a shadowing. A large area. You can't see it, but those specimens which will be shown up. So that's basically what we did for the prepare the sample for the electron microscope. That's what you see the phage. The bacteria, the flagella. This is the T4 phage. It's so clear because they freeze it and they cut it and they coat it with heavy metal. And this is what it looks like. You see the inside of the 3Ds, and all these places, they showed up after you do the heavy metal coating. Okay, so that's what we do preparing the sample. Scanning electron microscope looks like that. You have a vacuum system, photo multiplier, multiplier, scanning circuit, scanning oil, electron guard, and then you see the 3D images. This is what it looks like. The 3D images is very clear. The last one we want to mention to you, in addition to electron microscope, there is something more magic, is called atomic force microscope. Atomic force microscope, very typical, is using on the food surfaces. There is a laser, there is a cantilever, there is a photodiodes, and there is a scanner. They will generate these very beautiful colors. They will see the width, height, roughness of a bacterial surface. And they have a computer system called measure it. This is what I just did in the lab. And my graduate students did. It's so interesting. This is Listeria monocytogenes in a solution treated with peroxyacetic acid and hydrogen peroxide. Both of those agents is oxidizing bacteria, which means bacteria is overreacted during the metabolic process, so they burst. But we want to see how they look like. So you see the first picture? In bacteria buffering media, buffer peptide water, very organized. In the water sample, looks okay. These are the 3 pictures we using the treatment, peroxyacetic acid, hydrogen peroxide. We're going to talk about the chemicals behind it. Okay, what's the story behind it later on? Can you see with the increasing of the concentration, what happened? The roughness is increased. The roughness is decreased. At the end of, you didn't see any of the morphology. It looks like they screw together because they're getting killed. And you can't even measure, you can not even measure the width. And even the height is dramatically increased compared to these non-treatment. So this is clearly, you can see, this is a more organized morphology compared to using 0.5% the concentration of the antimicrobials, they get killed and damaged. So that's an atomic force microscope, the function for that. And we also could measure the width, the height, and the roughness of these, and you can see, and then you can do the comparison to see the statistical difference. And we obviously find width you cannot even measure. Height using 0.25, 0.5 or dramatically significantly higher than the other treatments, and the roughness is significantly decreased. So that shows you the bacteria, the foodborne pathogen, after treatments with antimicrobials, what the morphology looks like. Okay, so that's why I want to be introduced these things to you. Okay, so we finished talking about here. We're going to move on to the next section, which is we want to talk about the bacteria staining technology. So this is a very important, but honestly, we should have a lab. So we will be doing the gram stain at the end. Then we can explain everything to you very well. What is the story behind that, what it is, looks like. Okay. So let's remove these and we talk about the bacteria staining technology. So first the picture looks like this. Smear preparation. So we talk about this smear preparation. So first of all, when we talk about this smear preparation, we will actually mention about is simple stain. So what do you mean simple stain? Said very simple. I had a glass lines. This is the glass lines. And I'm gonna draw a circle. I'm gonna put a drop of water. And I'm gonna add a bacteria. I make a big smear. Then what we do air dry in the environmental temperature to 3 to 5 minutes. And this needs to be completely air dry, because otherwise what is going to happen? You will see your bacteria looks like this. Lots of the legs going to show up. That's not good. We don't want that. Then what we do? Heat fixing. So what's the function of the heat fixing? We would love to kill enzyme of bacteria and let it be attached to the surface. Very well. Okay. Then what we do? We need to add a dye. Dye is a chemical, showing you the color. Some of them have the toxication material there. So the first question, for the bacteria cell, why we can add a dye there? This is the reason is, if you have a bacteria, let's say E. coli 0157H7, let's just give an example. They grow at 35 degrees Celsius, 24 hours. What it looks like? They are negatively charged. And we can put a dye there, because all those dye is positively charged. You can see it right here. It's positively charged. Therefore, positive and negative electrons, they could be positive ions, negative electrons, they could be combined, attracted with each other. Therefore, they could be staying together. I'm showing you the color. So it's very simple principle behind it, why we can use a dye. So what are the dye we do? We have lots of basic dye. The most useful one is crystal violet. It is a purple color. We will mention this when we talk about Gramstein. And saffronin, we could use. This is a pink color. We also could use methylene blue. It's a blue color shade. We also can use malachite green. It is a green color, obviously. This is showing you the color, what it looks like. But at the same time, you also need to know, for some of the bacteria, we do not want to see the bacteria itself what it looks like. We want to stain in the background, then let it be showed up. So very interesting. So remember, in your biology class, you talk about F. Griffith's experiment in 1930s, talk about the DNA is a genetic information. He used a tool, which is a bacteria, Streptococcus pneumoniae. This Streptococcus pneumoniae is a diprococcal, have a very heavy capsule. So remember, we have a rough one, smooth one. The smooth one will be killing the mice, and the rough one not killing the mice. Blah blah. That's a story we don't talk. But from microbiology standpoint, that heavy capsule, we want to show it. Capsule is difficult to stain. So what we do? We need to stain the background. So how we do it? Bacteria is negative charged. Is that right? I want something also negatively charged as a dye. Because they can repel. This comes out with acidic dye, which is also negatively charged. This is called negative stain. But something I need to mention, we don't call this is a positive stain. We just say this is negative stain. What are the dye for negative stain? Nigrosin Indian ink. Stain the background, show you the color. Okay, stain the background and show out the bacteria. That's the positive and negative stain, what it looks like. Nigrosin Indian ink, Eason and Rose Bengal, acid diffusions, those for the negative stain. Now the positive dyes, there's a lot. But one thing I always talk in the lab, methylene blue does not have a very strong positively charge. And so I said, if you do not have a very good technology, please don't do it because it's not going to show up very good. So what's the function of these simple stain? We want to see the morphology of the bacteria, and we want to see the arrangement. So always remember, why we want to see the staining results is morphology of bacteria, which means the shape of bacteria. We also want to see the arrangement of the bacteria. So what is the morphology? It could be these dots, we call it a coccale. It could be these lots, the bacilla, we call it a lots. It could be like that, we call it a vibrio. Something else. What's the arrangement? It could be single, it could be double, duplo, coccale, It could be also 4 of them stacked together. We got a tetrade. It could be 8 of them stacked together. We call it a grip shape. It could be a chain like that. So on and on. That's called arrangement. Okay. Now, why it is important? Sometimes we could do a clinical diagnosis. You're not going to see the bacteria identification results. You have to give the treatment. So we need to do a simple stain to have the idea based on the symptom what it is. So if people have eaten ground beef undercooked, had a diarrhea, vomiting, nausea, what are we going to see? Could be E. coli, is that right? So E. coli you see using crystal violet, it's lots, short lots. If there is kids, immigrant kids from Mexico, and he's not getting any of the vaccination shots, when they have some called bone neck, and also have inflammation of the tonsil area. So we get a sample from the sputum or the tonsil area, what we see. It could be something like that, which is a club shape. That is called a corynebacteria. That could be corynebacteria. Then we know we need to treat antitoxin and antimicrobials at the same time. So this gives you a basic information. What's the morphology, the shape of the bacteria? Then you could determine preliminary, most likely, what type of the pathogen it is. And then, this is a very easy and obvious results for negative stain. This is Streptococcus pneumoniae. Look at how clear it is. Background is dark using Indian ink. You see the capsule there, and you see the Diplococcus cell right in the middle. So if you see the patient that looks like that, community-acquired ammonia, using antibiotics immediately, especially in the 4th semester season. So obvious, Streptococcus pneumoniae. We're going to talk about detail. Go to the exam too, okay?